Hello everyone, my name is Akanksha Nani, and welcome to my YouTube channel Neuro Chat with AG. On this channel, I plan to cover topics that are closest to my heart and support the neurodiverse community. Topics related to neurodiversity, autism spectrum disorder, and more. On the channel, I will interview thought leaders, esteemed researchers, scientists, professors, doctors, and psychologists, seeking their views and updates on their latest work, research, etc. I will also chat with parents of neurodiverse kids and adults and talk about experiences of neurodiverse individuals. Today we will be talking with Professor Henry T. Greely, known as Professor Hank, who specializes in the ethical, legal, and social implications of new biomedical technologies related to genetics, assisted reproduction, neuroscience, and stem cell research. Professor Hank chairs the steering committee for the Stanford Center for Biomedical Ethics and is also a professor of genetics at Stanford School of Medicine. In 2007, Professor Hank was elected a Fellow of the American Association for the Advancement of Science, receiving Stanford University's Richard W. Lyman Award in 2013, and the Stanford Prize in Population Genetics and Society in 2017. He published this amazing book, The End of Sex and the Future of Human Reproduction, in 2016. His other book, CRISPR People, The Science and Ethic of Editing Humans, was published in 2021. Professor Greely, welcome and thank you so much for coming.
My pleasure.
Could you tell us about your career journey and what led you to focus on bioethics law?
Sure. Time and chance. I was an undergrad at Stanford, and when I came to Stanford, I knew I either wanted to go into sort of science or government/policy. I wanted as a freshman to either win a Nobel Prize or become president or maybe Supreme Court justice, chief justice, or maybe both, maybe all three. Ultimately, I ended up majoring in science, but it was political science, in part because the second midterm in the third quarter of the FAST math sequence exposed that my native math ability had run out, and I overreacted to that, but also in part because I liked science, but there was no one science that really grabbed me. So I went to law school. I worked for a couple of federal judges. I worked in the Carter administration. I worked as a lawyer in Los Angeles. In 1985, I was offered a position on the Stanford faculty. They wanted me to teach oil and gas law because I had worked at the US Department of Energy. Now, oil and gas law is a tremendously interesting field. Stanford had had the country's two leading scholars of oil and gas law. They had both retired. Oil and gas law had nothing to do with the US Department of Energy. It was really a state law subject, but who was I to correct the Stanford faculty? So I said, sure, taught it for a couple of years, turned it into an energy law course, but along the way I had married a doctor. So I also started teaching health law. And in 1991, I decided I can't do both these. Each one is way too big, and combined, they're too big to the 10th power. So I made lists and lists of pluses and minuses. And I said, if I stay with energy law, the big issue for the roughly 30 years remaining in my career will be climate change. And I don't think we're going to have the political will to do anything meaningful about that in 30 years, in the next 30 years. So that will be depressing. But if I do health law, I think we're going to get a Democratic president in 1992. I think we're going to get universal health care. That'll be really exciting. So I chose health law. In retrospect, I actually think I was right about energy law. We didn't have the political will for 30 years, though thankfully my career has gone past 30 years, and I think we are now beginning to get both the tech movements toward tech solutions and the political will to maybe prevent my generation from leaving yours generation and future generations with a complete disaster. But on health law, things didn't work out quite the way I expected. The Clinton health plan crashed and burned. But luckily, and it was pretty clear there wasn't going to be any major health policy reform for decades. But luckily for me, the Human Genome Project started up. I had always liked science. I had read something about the Genome Project. I was the sort of person who buys Scientific American on the newsstand, but doesn't subscribe. I wasn't that committed. But I was put on the planning committee for a 3-day symposium Stanford was holding about the then new Human Genome Project. Met some of the scientists, really liked them, ended up giving a talk, met more geneticists. They started asking me for advice. And by 1992 or 93, I had found my career. I had found my calling at the age of 40, which was not, I don't think of it as bioethics itself, but as I like to call it law and the biosciences. Because some of it is ethics, but some of it is kind of straight law, like the patent rules regulating patenting biological materials. There are ethical implications to everything, but I like to do what I call law in the biosciences. I started with genetics. After Dolly was born, I did some cloning and embryonic stem cell research that led into embryonic stem cell research. 2002, I started working on neuroscience. Along the way, I started working on assisted reproduction, human subjects research ethics, human biological enhancement, de-extinction, and basically any bright, shiny object that caught my eye. So I think I have sort of academic attention deficit disorder, but I'm good with that. I like covering a broad range of topics, even though that does mean that in any topic I cover, there are people who know a lot more about the details of it than I do. It's both there's a plus and a minus. When you go broad, you lose depth, but when you go narrow, you've got lots of depth, but you lose the kinds of intersections and the kinds of insights you can get from knowing and comparing different things. Each strategy can work. I think it's really a question of which one fits your personality. And I have no doubt that I found the one that fit mine.
How do you see the role of law in the biosciences evolving in the next decade, especially with the recent advancements in biotechnology?
We currently know approximately infinitely more about biology than we knew 50 years ago. One of the things we know infinitely more about is there are infinitely more questions that we don't know the answers to about biology. One great thing about vibrant sciences is as they advance, you learn more new questions than you get new answers. But I don't think it's infinite. I think we are making progress in understanding biology in ways that can relieve human suffering, improve the environment, do good things. But that can also increase human suffering, further mess up the environment, and do bad things. And one of the roles law has many roles in this, but one of them, along with some other fields, is to try to tilt the scale so that we get more of the benefits and fewer of the harms. So on the one hand, I think there is there's already a substantial and will only increase demand for lawyers who know something about biology, because biology is a big business and it's going to get to be a bigger business. At the same time, I think there is a large and will be growing demand for people knowledgeable and interested in law and biology to talk about the policy implications, whether it's in government, in business, or in academia. So it's got a big role. It's not the only player, but it is a very important player in how we decide collectively and individually what to do with the possibilities that advances in the biosciences bring and how we decide to do it.
And what do you think some of the most pressing ethical issues in genetic engineering are today?
Is it safe? Is it effective? Those are always the two most important ethical issues in anything. We often don't think of them as ethical issues, but they are deeply ethical issues. It's deeply unethical to do something to someone that is unreasonably unsafe or to sell somebody something that isn't going to work. And those are huge issues. So, for example, right now, one of the hot questions is the use of what's called probabilistic risk scoring. There's some other names for it too, polygenic genetic screening in human reproduction. We know that there are some conditions, diseases, traits, conditions that are powerfully genetic. If you were born with more than 40 CAG repeats in your Huntington gene, the only way you're not going to die from Huntington's disease, as far as we know, is to die first from something else. And if you have fewer than, say, 37 repeats, there is no way that you can ever get Huntington's disease. That's powerfully genetic, and that shows up easily in testing, and people use that to try to make decisions about their careers, their life plans, or whether to have children, and sometimes which children to have. Almost every disease, probably every disease, has some genetic component. But for most diseases, it sort of falls into three or four categories. First, there are going to be a few people who've got an overwhelmingly strong genetic connection to whatever the condition is. So, for example, with Alzheimer's disease, about 1 person in 1,000 inherits a mutation in something called PS1, presenilin 1. That means unless they die first from something else, they will die from early onset Alzheimer's disease, usually in their 40s or 50s. They make up about 1% of people with Alzheimer's disease. About 25% of people have one copy of a version of a gene. The gene is called ApoE, apolipoprotein E. There are three main versions called ApoE2, 3, and 4. So now ask me a question.
What does having each gene result in?
Wrong.
Yeah.
Question is, what happened to ApoE1?
Oh, okay, yeah.
When you name things 2, 3, and 4, that's a lot. And it turns out to be kind of a meaningful question. ApoE1 was the first variant noticed, but it was really, really, really strongly negative, not for Alzheimer's, but for a bunch of cardiovascular stuff. And really, really strongly negative genetic variants tend to be rare. Natural selection does not like them. So ApoE1 was the first to be noticed because it was so powerful and so bad, but that also meant it was rare. So you and I undoubtedly have 2 copies of ApoE, one from our mom, one from our dad. We either have 2 copies of 2, 2 copies of 3, 2 copies of 4, a 2 and a 3, a 2 and a 4, or a 3 and a 4. 6 combinations, right?
Mhm.
If you have 2 copies of ApoE4, it looks like your risk of being diagnosed with Alzheimer's is about 80 to 90% if you live to your 80s. That's a lot higher than the normal 10 to 15% rate, but it's not 100%. We don't know why. And that's about 4% of the population. If you've got one copy of ApoE4 and one copy of ApoE2 or 3, your risk is doubled or tripled. But it's not yet 50%. It's 30 or 40 or 45%. We don't know why. If you've got 2 copies of ApoE3, and that's about half the population, about 20% of the population has one copy of ApoE4, about 4% has 2 copies, about 50 to 60% has 2 copies of 3, well, your risk is average. If you have 2 copies of ApoE2, that's 1 to 2% of the population, your risk of Alzheimer's looks like it's 0. You're at higher risk for some things, but not for Alzheimer's. So ApoE has something to do with Alzheimer's disease. It can either increase your risks, but not to 100%, not like Huntington disease or early onset Alzheimer's disease in PS1. It can increase your risk substantially. It can decrease your risk somewhat. If you're a 2-4, your risk is about half normal. It can decrease your risk entirely. But for all that, more than half of people with Alzheimer's disease have neither a PS1 mutation for early onset Alzheimer's nor an ApoE4 allele. Is their disease genetic? Well, there are another 100 or so genes that have been associated weakly with Alzheimer's disease. but only really weakly. So things like asthma or schizophrenia, or the variety of different things that get lumped into the category and called autism. There are some aspects of each of those diseases that are powerfully genetic. There is something called Timothy syndrome, which is a rare version. It's a rare kind of a rare thing in the cluster of things that is called autism. It's like 1 in 50,000 births. It looks like it's completely genetic. It's really severe and huge intellectual deficits. People don't learn to talk. But there's seizures, and that's completely genetic. But looks like there's a whole bunch of other genetic things that maybe slightly increase your risk or slightly decrease your risk, but it's only slight. And we can say, well, if you've got this variant, your risk is a little bit higher. And if you've got this variant, your risk is a little bit higher. But we really don't know if you've got both of those variants. Does that make your risk much higher? Does it make it the same? Does it make it even lower? We don't know. So this is a law professors usually have 50-minute hours, and so answers tend to be lecture length. This is a long intro. There are companies that are currently willing to sell people risk information based on statistical manipulation of large data sets of small risk sets, of small risk variations. And they will say, well, if you've got these variations, your chances of this are double. And if you've got these variations, your chances of this are halved. But it's looking at 100, 200, 1,000 different variations. And they're selling it for, among other things, intellectual ability. But we don't know whether that's right. This is scientifically quite controversial, at least when it applies to individuals. You can say over large groups, yeah, that risk is higher, that risk is lower, but within any individuals who have, after all, 6.4 billion base pairs of DNA and lots of variations, nobody has ever had your exact DNA before or ever will, including an identical twin. Because the moment you and your identical twin separate, you begin to mutate and get slight differences. We don't know whether this works. So that to me is a big scientific but also ethical question. Should we be using for human reproduction or human health these probabilistic risk scores based on hundreds and thousands of small pluses and minuses to risk caused by variations? That's a hot topic right now.
In terms of legal framework, with all these new technologies like advances in CRISPR and other gene editing technologies, how do legal frameworks stay updated with all these changes that are happening?
Not very well, but better than you might think. The real answer is it depends, which is the real answer to any question you ask about law or pretty much anything else. One of the big things it depends on is where are you talking about? The UK has a pretty strong regulatory system for at least reproductive uses of genetic information. They've got something called the Human Fertilization and Embryology Act, which created the Human Fertilization and Embryology Authority. And they exercise pretty tight, I'd say too tight, but pretty tight control over what can be tested for when it comes to prenatal testing or it comes to pre-implantation genetic diagnosis. US has none of that, which I think is too little regulation. So in the US, anybody can test for anything they want, especially if they do it through a licensed clinical lab. They don't have to prove to FDA or anybody else that their test works. They may be liable for medical malpractice, but they don't have to prove that their test works. There's no regulation of that. And not everything is law. So there is the American Society for Human Genetics, and they put out guidelines about what they think should and shouldn't be tested for, but those are voluntary guidelines. No one's required to follow them. There's the American Society for Reproductive Medicine. They also put out guidelines for preimplantation genetic diagnosis and what should and shouldn't be done. Again, voluntary guidelines. It's better than nothing. But I think maybe there should be some regulation, though doing the regulation would be tricky. Having said that, we do have some regulation, but it's haphazard and almost like arbitrary and capricious. So you cannot legally CRISPR human embryos to create a genetically edited person in the United States, because the Food and Drug Administration has taken the position fairly consistently since about 2000. And when the birth of Dolly birth of one sheep led people to be terrified about armies of cloned warrior slaves. FDA has taken the position that a genetically modified human embryo, if used to make a baby, intended to be used to make a baby, is a drug or a biological product and cannot be legally done in the United States without FDA approval of it as safe and effective. So you'd have to get FDA's permission. FDA has always made it pretty clear that they're not likely to give permission unless you've got lots and lots and lots of really good evidence. Nobody's ever asked. However, in 2015, members of Congress added on an amendment to an appropriations bill. Appropriations bills have to go through every year. They only last for 1 year. And that's why every few years, usually it's the Republicans cause the government to shut down because they refuse to pass an appropriations bill or what's called a continuing resolution, which is another way of giving government agencies money, giving them the authority to spend money. Appropriations bills are kind of special because when you're getting down to the wire and time's getting tight, they need to be passed. So they become what are called Christmas tree bills, where there's the main bill, there's the tree, which gives government agencies lots of money, but then there are all these weird little ornaments hanging off the edges of them called riders. Since 2015, every appropriations bill that's governed FDA has included a rider that says FDA may not consider for approval any application to do research that would implant a human embryo with heritable genetic modifications. So trying to CRISPR babies in the US is illegal because trying to use a drug or biological product, which FDA says the embryo would be, that hasn't been approved by FDA is illegal. FDA could approve it, except Congress has said, FDA, you're not allowed to even consider approving it. But every time Congress says that, it's for 1 year only, and they have to keep repassing the bill. That's a strange way. Oh, and it never really gets hearings. It never gets much consideration. That's a strange way to do public policy, especially since there's another process called mitochondrial transfer, which does put in inherited changes. You probably learned about the mitochondria and the powerhouse of the cell. It's always the powerhouse of the cell. Why it's not the dynamo or the generator or the battery or whatever, but it's always the powerhouse of the cell, shaped like a kidney bean, or a kidney, which is shaped like a kidney bean. Mitochondria are weird. They have their own DNA. So in the nucleus of your cells, presumably you have 46 chromosomes, although close to 1% of us have either more or fewer than 46 chromosomes, but most of us have 46 chromosomes, 22 autosomes, cleverly numbered 1 through 22, and an X and a Y. And I'm guessing, looking at you, that you have an X and a Y. Women usually have an X and an X, although in biology, nothing is always true. So there are biological women who have an X and a Y and biological men who have an X and an X, because what matters isn't the whole chromosome, but the genes that are on it. Anyway, that's 6.4 billion base pairs, billion letters. Have you ever read The Lord of the Rings?
Yes.
Whole thing?
Like three main books.
Okay. The Hobbit too?
Yes.
Okay. So if you think of every character, not person, but every letter, punctuation mark and space in The Lord of the Rings plus The Hobbit as one base pair of DNA, you have in your cells, in each cell that has human DNA, you have the same number of characters as roughly would be found in 1,000 copies of The Lord of the Rings and The Hobbit. 6.4 billion is a big number. People don't really understand how big a number that is. 1,000, you'd have to read it 1,000 times. And that's your genome. That's only got 4 letters. And there's a lot of stuff that's harder to understand in the Elvish songs, but it's long. Mitochondria have completely different DNA, one little circular bit of DNA that's about 16,000 base pairs long. 6.4 billion, 16,000, and yet there are some diseases that are caused by errors in the mitochondrial DNA. Eggs have, I think the technical term is a boatload. There's another version of that technical term. They've got a lot of mitochondria. Sperm basically have no mitochondria that enter the egg. The sperm's mitochondria are stuck in their tail and they don't go in. So every baby has mitochondria only from its mother. If the mother has a genetic disease caused by a mitochondrial defect, the baby will. Almost always, because nothing in biology is certain. Somebody said, starting 25 years ago, why don't we take mitochondria, healthy mitochondria, from women who don't have a mitochondrial disease and match them in an egg with the nucleus and the 6.4 billion nuclear base pairs of DNA of a woman whose nuclear DNA is fine, but who's got this mitochondrial disease, put them together, either by adding the healthy mitochondria to an egg cell from a woman who wants to have babies, has bad mitochondria, but a good nucleus, or taking a egg cell from a woman with good DNA, good mitochondria, taking the nucleus out and putting a nucleus from the woman who wants to have babies but can't because of her bad mitochondria in there. That's been done. It seems at least grossly safe, but it's never gone through FDA approval. It's Greece, Turkey, there are a number of countries Ukraine used to before it got busy with, before it was grossly violated and invaded, have done mitochondrial DNA transfer. You can't do it in the US because it creates a heritable genetic change. And so, although that rider that stops CRISPRing babies was intended to stop CRISPRing babies, it's also stopped mitochondrial transfer research in the US. So, yeah, we kind of regulate this stuff, and maybe we don't do it in the worst possible way, but we certainly do it in a bad way.
Yeah, thank you. That's a really huge number. So, what are your thoughts on personalized medicine to transform healthcare, like in autism and for the larger community?
So, I'm not a specialist in autism. I don't know much, picked up, I know some. One of the things I know is it's almost certainly not a single condition. It's a whole bunch of conditions, probably with different causes and different mechanisms of action that get lumped together because they have similar symptoms. Schizophrenia is like that too. So you get diagnosed as schizophrenic if you have 3 out of 7 symptoms listed in the Diagnostic and Statistical Manual, 5th edition. That means 2 people can both get a schizophrenia diagnosis without having a single symptom in common. So schizophrenia, autism, almost certainly lots of different disease or lots of different conditions, whether you want to, some of which I would term diseases, some of which I wouldn't term diseases, depending on their severity. I think that so I know something about it, but I don't know that much. I think personalized medicine is a wonderful thing. My wife retired after 30 years as a pulmonologist, and she always did personalized medicine. She did not do the same thing to an otherwise healthy 20-year-old with a bad cough as she did to a 94-year-old with 4 organ systems in failure with a really bad cough. Personalizing medicine is what having doctors is for, and nurses and other healthcare workers. What you mean, and what's been, the way it has been taken over, means genetically personalized medicine, which I think sometimes can do great things. So for a few people, not very many, but a non-trivial percentage of people with bad cancers, picking based on genetics of the cancer, not of the underlying person, but of the tumor, the mutations in the tumor from that person's original DNA, you can pick the right drug and it works great sometimes. If you know that a kid has no good copies of the gene that is, when mutated, when ineffective, causes phenylketonuria, you can do personalized medicine by putting that kid on a really, really low phenylalanine diet, which is not fun and easy, but it keeps that child from having an IQ of about 30 and gives them sort of a normal IQ. That's personalized genetic medicine. Gene therapy is personalized genetic medicine, and about, depending on how you count, 10 or so gene therapies have been approved, and probably fewer than 100 people have received them so far. Again, it depends in part on whether you count the CAR T cancer therapies as gene therapy or not, and there's disagreement about that. Cystic fibrosis, about 30,000 Americans have cystic fibrosis. Used to be a death sentence. Used to be people with cystic fibrosis would not live past 10. But for the last 40 or 50 years, the treatments have gotten better, and they've gotten better about a decade every decade. So then it became you died in your teens, you died in your 20s, you died in your 30s. Now the mean was you died in your 40s. A couple of years ago, a company did a small molecule drug. It's not a gene therapy. It's not an RNA interfering treatment. It's not CRISPR. It's nothing sexy. It's an old-fashioned small molecule drug. And they now come up with 3 versions of it. And it basically cures the symptoms of cystic fibrosis in 90% of patients. Well, that's a personalized therapy, it's personalized medicine, and that you only give it to people who've got cystic fibrosis. And you only give it to the 90% of people with cystic fibrosis who've got the particular mutations it works on. That's personalized medicine. Do I think that getting everybody's whole genome is going to let us live forever? Live to be 150? Banish all disease? No. Most diseases are too damn complicated. Mixes of genes, and not 1 or 2 or 3 genes, but tens or scores or hundreds of genes, and environment and chance. So I think it will be a good thing. I think, like everything else in our current society, it gets overhyped. And it's gone through a couple of waves. The hype cycle is like a sine wave, It's gone through a couple of cycles of the hype cycle. It first started out with the ugly name pharmacogenomics in the mid-1990s. Then it became personalized medicine. Some have now tried to change it to precision medicine. And it's done some good things. It will do some better things. Is it going to do everything? Not even close. But it's worth doing. In autism, I have no idea. I mean, I don't know enough about it. I do think that autism is going to be peculiarly hard because anything involving brains is really hard. Brains make everything else look simple. And neuroscience is approximately 2 orders of magnitude harder than genetics, which turns out to be pretty hard. And there's so many different things that get called autism or autism spectrum disorder or Asperger's or all those different labels. We don't know how many different conditions there are, how many different traits there are. We don't know what causes hardly any of them. We don't know how to change hardly any of them. So we will learn. But my guess is with the whole spectrum of things that gets labeled as autism or autism spectrum, it's going to be slow, and frustratingly slow for some, happily slow for others. So if you're worried that pretty soon parents are going to be able to abort any fetus that would have autism, I don't think you need to worry about that. Because I don't think the science is going to be anywhere close to that. They might be able to abort any fetus that would turn into a child with Timothy syndrome, because that does look like it's one very powerful gene. That's like less than 1 in 1,000 people with an autism diagnosis. And that is that's a really terrible condition. And lots of autism ranges from, I don't want to say this, as good as or better than non-autistic people to just completely awful. Constant seizures, no huge developmental disabilities. Those things like that both get called autism. But I think both the science and the ethics of them is going to be quite different. I have no problem with parents deciding they don't want to have a child with Timothy syndrome. I would have a big problem with parents using probabilistic risk scoring to say, yeah, we don't want to pick this embryo because it has a 3 percentage point higher risk of having something somewhere that maybe is somewhere on the autism spectrum. Parents avoiding having a child with Timothy syndrome is enormously different to me than parents making a decision not to try to transfer for possible implantation in embryo where polygenic risk scoring, probabilistic risk scoring has said it's got a 3 percentage point higher risk of having something somewhere on the autism spectrum, both because I think the science is likely to be bad and because I think a 3% risk of having something somewhere on the autism spectrum isn't necessarily a bad thing. Having Timothy syndrome, I think, is a bad thing. Having a lot of the things that get called autism is not necessarily a bad thing.
In a session of yours, you talked about interventions in Parkinson's and Alzheimer's disease. Are there conflicts in ethics on research versus its implementation, especially in recent surgeries on humans to treat these conditions? You know how you can do a testing to kind of see if you're gonna end up having if like the amount of tau or amyloid beta in your brain is increasing.
Yeah.
So, and then people might take drugs to try and flush out the protein plaques out of their brain. So,
Right.
is there a conflict in ethics on that kind of stuff?
There are ethical issues there, both with respect to the testing and with respect to treatments. And with treatments, with respect to the research and treatments and to the use of treatments themselves. So let me start with research for treatments. We do not allow human subjects research where the harms greatly outweigh, where the likely harms to the patient, to the subject, to the research participant outweigh the potential benefits to the subject or to science or medicine, even if the subject agrees. We also generally don't allow research on people unless they've given informed consent. But even if you give informed consent, if somebody wanted to do research on me that involved drilling a hole in my skull and pouring benzene into it to prevent Alzheimer's, even if I said, sure, I'll go for it, what do I have to lose? That wouldn't be allowed because the risks are too great and the potential benefit is for something like that zero. So yes, we have some ethical limitations on what research can be done with humans. We also have ethical and legal limitations on what treatments can be given humans, which is basically FDA has to have agreed that it's safe and effective. FDA has agreed so far that 3 drugs that purport to treat Alzheimer's are safe and effective. Personally, I disagree with the FDA on that, but the FDA makes the law, I don't. Right? The FDA implements the law, I don't. But even the FDA agrees that there are some significant risks and the benefits of these drugs seem quite small. But it's something. And for a community that has never had any useful drug. Then, so in terms of that, yeah, there's some ethical concerns. In terms of testing, there are also ethical concerns. mainly about the consent, whether the person who asks for, well, two. One is whether the person who's asking for the testing has had a good opportunity to make a fair decision, has been informed about the pluses and minuses, and has a chance to think about it and has a chance to ask questions about it. A lot of people want to get tested for genetic disease. A lot of people also don't want to get tested for genetic disease. Both sides, neither side is right nor wrong. It kind of depends on you, your life circumstances, your personality, your goals. One of the really intriguing things is a lot of people who learn they have a 50% chance of having Huntington disease, which is just a terrible disease. It strikes people in their 30s and 40s, and you take about 10 years to die, losing muscle control and then emotional control and then cognition. Terrible disease. But a large percentage of people who learn that they are at risk choose not to get tested to find out whether they're going to get it or not. They're not crazy. That's the choice they want. So one thing is to make sure that when people get genetic testing, they've had a fair chance to know and think about what they're getting. And the other is to make sure that the test results don't get misused. They don't get used to discriminate against them in health insurance or employment or other things. In the US, we have the Genetic Information Non-Discrimination Act, which federal law prohibits genetic discrimination based in health insurance or in employment. But it doesn't cover life insurance or car insurance or adoption or a bunch of other things where people could, in theory, be discriminated against. The good news is there doesn't seem to be much genetic discrimination going on, in part because genes usually don't, for 5% of people, they tell you a lot. And for 95% of people, they don't tell you very much at all. But also because I think all the insurers and others realize this is a politically very sensitive topic, and they've been walking on eggshells.
Okay. And speaking of like genetic testing and how you mentioned how some people choose to get it done and some people don't, I think like taking that to the next level. So if genetic selection becomes available to the public,
It
do you think,
is already.
I guess what I'm trying to get at is like, higher level. Do you think it should be restricted for medical reasons? Only people with a certain family history.
So that's a really good question. You're talking about testing or the use of the test to, say, pick embryos or pick fetuses?
I think both.
Okay. So I'm in favor of restricting it to tests that have been shown to be reasonably accurate. And right now, there's no requirement in the US of proof that the test works. There are requirements of proof that the lab does the test well, but that's different from proving that the test works. I could say I'm going to predict whether you'll get Alzheimer's disease by flipping a coin, and they can prove whether I flipped the coin well and whether I truly get a random 50% plus or minus response, but didn't say anything about whether the coin flip actually proves anything. So I do think there should be proof of the reasonable accuracy of the test before it gets used, either for a test or before the test results get used. It is tempting to try to separate diseases from non-diseases. A problem with that is there is no great, nice red line in nature that says this is a disease and this is not a disease, as I think lots of neurodiverse people have argued. And gays and lesbians were, until about 1980, listed as having a mental illness in the Diagnostic and Statistical Manual of Mental Illness. So, what's a disease and what isn't varies. I think that different societies will make different choices there. And as long as the science is good and the people who make the choices are well informed, and we're confident that it's safe and it's widely accessible within the population, all of which are big ifs and none of which you can take for granted, then I probably don't have a problem with it. In part Now, this is in part informed by a deep skepticism about how much genetics is going to be able to tell us. I think it will be able to tell us what what an embryo's hair color would turn out to be. Right now, it can say light or dark, but it's not much better than that, except for really red hair with freckles. That's a well-known single gene issue. Everything else, we're okay. We'll get better. Things people mainly care about, personality traits, behaviors, those are really, really hard. And I don't think we're ever going to get very good at them, which makes it easier for me to say, yeah, if it works, let people test for IQ, whatever that means, because I suspect at most they'll be able to say has a 10% chance of being above average, significantly above average, and a 10% and a 5% chance of being significantly below average, and an 85% chance of being average. Not very useful.
This is something I'm pretty curious about, like your take on this. So, what future advancement in biotechnology are you most excited or concerned about?
That until this April, I thought was, yeah, it's going to happen, but it'll be 30 or 50 years away, and I don't have to worry about it. But lately I've been thinking, this could happen within 10 years. And it's one I have mixed feelings about. It's full human ectogenesis, making babies completely outside of people. The classic scenarios for this, going back to Brave New World in 1934, with its babies in the hatchery and bottles, was either a machine that would take the place of a uterus, or and more recently, build a uterus. We will learn using stem cells how to make hearts and livers and kidneys. We're trying. We'll do those first because they're life essential organs that are in short supply for transplant. So people are dying. That'll be 20 years away before we can figure out how to do that because most organs are complicated. But once you do that, somebody's going to build a uterus. And you take a uterus and you put it in a box and you hook it up to a machine that feeds it blood that has the right oxygen and the right sugar and the right hormone levels. And it goes through a heart-lung machine and a dialysis so that the blood is kept good. And you put a human embryo in it and you wait 9 months. That's what I call, where do you get, that would be full human ectogenesis. A baby that from the moment of fertilization until the moment of it was declared full term was never inside a person. That I still think is 30 to 50 years away. But I've recently realized that uterus transplantation has gone a lot faster and farther than I thought. And something called normothermic machine perfusion, keeping organs alive outside the body by pumping blood or other fluids through them at a body temperature, has also gone a lot faster and farther than I thought. So I'm working on writing something about what I call donor uterus ectogenesis. You get a donated uterus, donated with informed consent, either from a cadaver, where the consent, of course, is before the cadaver became a cadaver, or from a surgery, and you hook it up and you keep it alive, and you put embryos in it, and you make babies. Now, I think it's at least 10 years of research before one could, in good conscience, even think about trying that to make real babies. But it's 10 years instead of 30 or 40. And first, you would try to see how long you can keep the human uteruses alive ex vivo outside a body. And the good news there is, even if it doesn't work to make babies, it could be a really great research tool to study things like endometritis or other miscarriage issues. And if it looks like it's working, then you start taking uteruses from non-human mammals and you see if you can keep macaque uteruses alive ex vivo. And then you see if you can make baby macaques that way and you see whether they're healthy or not. And people have talked about ectogenesis for decades. You can even trace one version of it back to the Bhagavad Gita, which I never pronounce right. But there's a story in there about a queen who manages to make people out of butter, purified butter, ghee. Um, but it's always been hypothetical. And there's a very strong discussion with some people saying, no, this would be terrible. Ectogenesis would make women unnecessary. This would be terrible. It would be exploited by men. And there are feminists who say, no, this would liberate women. They no longer have to be pregnant. And pregnancy is not not an entirely benign procedure. It's uncomfortable and it's risky. And it takes you out of the workforce for a while, and it, means you can't have a glass of wine without people staring at you. Um, so I don't think, even if it worked, I don't think it would be the end of the world, but I think it would be really interesting. And I don't really know yet what I think about it, if it worked. But of course, as I told you earlier, the two most important ethical issues are always, is it safe and is it effective?
And are there any big, I guess, international agreements that you think are necessary to address these global bioethical issues?
God, I hope not, because international agreements are very rarely very effective. Hard to get countries to agree on things. One way to get them to agree on things is to make them so vague that the country really can do anything at once. Another way is, yeah, countries will agree to it, but not enforce it. I'm not optimistic about a strong role for international agreements. It's not to say they couldn't play some role, but in a way, we've already got a really important one. It's not strong enough, and that's the Chemical and Biological Warfare Convention. I mean, if you really wanted to ask me, if I really wanted to tell you what scares me the most about the new bioscience, it's biological warfare, which now, in theory, could be done by two kids in a garage, thanks to CRISPR. And yeah, we've got a convention against it, an international treaty against it, but I don't put much weight on its effectiveness, especially if it's two kids in a garage.
What advice would you give to young lawyers and bioethicists who want to work at the intersection of law, ethics, and biotechnology?
Do it. Do it. It's important. It's fascinating, and it's just so cool. And biology is close to infinitely complicated, in part because it's not engineered, it's not designed, it's a product of history and chance. Law and societies are almost infinitely complicated and fascinating because they're not designed. They're products of law, of time and chance and evolution, just like life is. So you've got these two really complex, fascinating things that are important to each other. And to be able to even just watch, let alone try to influence what happens when they come together is it's a great privilege, but it's also just enormous fun. Fun is good.
And would you like to share anything else with our audience about the future of law, bioethics, or biotechnology?
It's got one. I hope it'll be a good one, but whether it turns out to be on balance a good one or a bad one depends on decisions that get made by potential parents, by doctors, by legislatures, by judges, by all sorts of different groups. And I do my work in the hopes that the better they understand it, the better the chances are that they'll do more good than harm.
Um, Professor Greely, thank you so much for, doing this interview. I really did learn a lot that I didn't know. Yeah.
Good. Well, as you may have noticed, I like to talk, so thank you for giving me an opportunity.